Thanks. Thank you very much for the invitation to speak. I'm actually recording this and I'll put it up on YouTube so all the people who aren't here can experience it, and if the screen does get too bad, it seems to not so bad now, that smear, but if it gets out of control you'll be able to watch it again in glorious Technicolor. So what I was going to do, I've just got a couple of slides to talk about microbiology in Birmingham, because that's part of the reason for being here, is just to explore potential collaborations and so forth, and let you know about the changes that are going on. Then I'll talk about high-throughput sequencing, look at its applications in clinical microbiology and focus on 2 case studies. And then at the end I will say some hopelessly provocative things about the future of microbiology as I see it. So in Birmingham we like to think we've got a critical mass of researchers, funds and facilities. I would say probably in the top 3 to 6. Some of my colleagues say, oh, we're the biggest now. And I said, well, you know, how can you actually know that? And what do you count as a microbiologist, and what do you count as a PI and all that? But we're doing okay, I think. And then in terms of spectrum, we look at model organisms through to translational research. I think we're early adopters of technologies like omics approaches. And we've invested in microbiology through as you pointed out, Tim Mitchell's come, we've had a drug discovery unit set up, we've got this Birmingham Fellows Initiative. And then most recently we've set up an Institute of Microbiology and Infection, which is there to consolidate research that's going on across the campus, and particularly research going on in 2 colleges, College of Medical and Dental Sciences and the Life and Environmental Sciences. And we've actually co-located the bacteriologists in one building. And we got £200,000 out of the University Centre as start-up funds. There are some oddities here because, although we're calling it microbiology infection, it applies to bacteriologists and the virologists to have their own sort of separate way of doing things at the moment. And that's an issue for us to address in the future, I think. Now this, I'm not going to speak through this because that's not what I was here to talk about, but this is just to show you the breadth of people and the kinds of things we do, and I'm not going to read it out. You can scan over that quickly, but you can see there's lots of people doing lots of different kinds of things within our broad umbrella of microbiology. So that was all I was going to say about the Institute for now. I'm happy to talk about it in questions if people want to. Well, what I'd like to do is actually talk about some research and look at an area which I find particularly interesting, which is high-throughput sequencing. And I see this as a new opportunity. It brings sequencing, and this slide is a bit old now. I mean, we're probably talking 10,000 times faster and 10,000 times cheaper than what it was a few years ago with conventional Sanger sequencing. And this might be called what economists dub a disruptive technology, because it's basically a game changer. It changes the landscape of opportunities, and you have to rethink what is possible because of this technology. Now, we've just had a review out in Nature Reviews Microbiology, and I'm not going to go into a lot of the technical details of how sequencing works and what platforms and you should use and so forth. If you're interested in that, look at that review. But the key point is that this relies on fundamentally new approaches that are quite distinct from what went before in terms of Sanger sequencing. And a key point is that there are several technologies in the marketplace, and there is lively competition between them. And in a sense, this creates what we might call a permanent revolution. If someone comes to me and says, I'm writing a grant and I want to do some sequencing in the 3rd year, which will be like 4 years from now of the proposal, what will it cost? And what can we do? I just throw my hands up and say, I have no idea. All you can do is cost it at today's prices, because we have no idea what the landscape will look like in 2, 3, 4 years' time. In fact, some people have used the term hyper-Moore's law. So people use the term Moore's law in computing to say that the performance of your laptop will double every 18 months in terms of hard drive space and speed and so forth. If we look at what's actually happened this is just looking at cost, but similar things in terms of throughput and so forth. If you look at conventional sequencing, Sanger sequencing, it was obeying Moore's law pretty much in the early part of the millennium. And then around 2007, 2008, the shape of the curve changed dramatically. And we're now probably doubling every 6 months or so. And if you project this forward, where do we get? Well, I would say that we can reasonably predict that we will actually reach what we might call a technological singularity, the sequencing singularity, where sequencing becomes so cheap and so easy that it becomes the preferred way of doing almost anything you can think of. You hear people say, oh, you can't do sequencing, it's too expensive. Well, you know, maybe now, but you come back in 5 years and the thing will be changed. In fact, I read one of the papers from here. There's a guy who does this electronic nose, and his opening line in his paper was, you could do all this by sequencing, but it's too expensive. Well, you know, I'll take a bet with him. Come back in 10 years and we'll see which technology is being used. There are many advantages with high-throughput sequencing in sort of the broad picture of microbiology and diagnostic microbiology. It's kind of open-ended. You just can sequence stuff and not know what you're going to find. So in the past, people have used molecular methods in microbiology, things like PCR, but there you have to know what your target is and design primers specifically for that target. Whereas if you just sequence the DNA, you reveal that horrible Rumsfeldism, the unknown unknowns, the things that you weren't expecting to find or didn't even realise might be out there. Because it's universally applicable, in the field of microbiology, you know, all agents have DNA or RNA with the sole exception of prions, and I guess that's the only exception, really. And in terms of resolution, once you've got down to the genome of an organism and sequence that genome, there's nowhere further to go in terms of resolution. This is the ultimate. And you could argue that this will replace all other existing typing methods for assigning evolutionary affiliations and so forth. Another interesting development in just in the last year, 18 months, is that there's a whole new crop of sequencing platforms that we term benchtop sequencing platforms, which are effectively about the size of a laser printer, as their name suggests. They just fit on a benchtop. And these have the ability to generate data quickly and cheaply, and in terms of setup costs also are relatively straightforward and simple, so that they can have an impact on real-world clinical epidemiological problems in normal environments. You don't have to be a large sequencing centre anymore. You can just put one of these into your setup and it works. Now, this is a rather busy slide. I've just taken a figure from that review that we had, and it just illustrates there is a lot of variety out there in terms of workflow. Most of the platforms currently have a number of steps that are in common, but there are variations on a theme within each one. So you have template DNA. You have to have a certain quality and quantity of that. You fragment that, and then you tag the fragments, and then you amplify, and then you sequence. Now, that applies to most of the platforms, but there are platforms already out there, PacBio in particular, that doesn't require that amplification step. It's called single molecule sequencing, and there are, as we'll see later, there are possibilities on the horizon for that as well. But basically, this is the way it works. You have this fragmentation tagging, amplification, which can either be on beads, on a solid surface on a bead, or on a two-dimensional surface in the so-called bridge amplification in the Illumina platform. And a variety of different instruments down there. I don't have time to really go through all of them. This is, again, a cut-down version of a table in that review. And, again, it's very busy. I'm not expecting you to read all of that. But it's there just to remind us that actually we don't have a perfect platform at the moment. They vary in terms of runtime, throughput, price, read length, and there's always a trade-off to be had when you look at these different platforms. And we are waiting for the perfect platform for these kind of applications, but we're not quite there yet. Now, in terms of genomic epidemiology for bacteria, in the last few years, we've seen this thrust itself onto the scene in terms of a number of papers. I've just put up a handful there. We're probably now talking, I would guess, somewhere between 2-3 dozen papers in looking at whole genome sequencing as an epidemiological tool in bacteriology. One of the earlier examples there was in the investigation of the Amerithrax incident in the US, where there was deliberate contamination of the US postal system with anthrax bacilli. High-throughput sequencing contributed to identifying the source of that particular stock of anthrax. It wasn't the only way in which it was identified, but the genome analysis did help. In fact, Paul Keim, who's one of the authors on that paper there, once told me that he believed that he had delayed the first Iraq war by 6 months or so because he was able to tell the US administration that the strain in that outbreak was actually a homegrown lab strain. It wasn't something coming from Iraq. Even more political than Amerithrax was this investigation of what went on in Haiti with cholera. When the genome was sequenced of the Haitian cholera outbreak strain, it turned out to be much more closely related to similar strains from Asia, from South Asia, than to anything from the Caribbean or from South America. And the finger was immediately pointed at Nepalese peacekeepers that had been brought in by the UN. And in that first paper there, that was kind of put out as a tentative conclusion, but the evidence was firmed up quite considerably in subsequent investigations. And that had political kickback in Haiti, obviously, created some problems. In terms of more kind of focused on normal infection kind of stuff that people look at in hospital infection, here's a recent paper here. I'm looking at Staph aureus and C. diff using benchtop sequencing. Now I'm going to draw on my own work looking at 2 case studies, 2 different organisms now. And one organism we fixed on as an interest a few years ago was Acinetobacter baumanni. Now this is a gram-negative bacillus, and it's particularly notable because it's conventionally multi-drug resistant. And in fact, it is resistant to so many things that typically you only have a couple of agents that you can use. And these agents, colistin is a very old agent that people didn't like to use because it's quite toxic, and tigecycline is a very new agent. But in fact, there are isolates now that are resistant even to those. And this is moving towards pan-resistance, where we're kind of running out of antibiotics. It's associated with wound infections, ventilator-associated pneumonia, bloodstream infections, and it's been a particular problem in returning military personnel. We've seen that both in the UK and in North America. And in Birmingham, on the Birmingham campus, all the military personnel casualties from overseas get repatriated there. And so we get the soldiers coming back from Afghanistan. And one problem that's been seen both on our campus and has been reported from North America is that you get transmission of this agent from military to civilian patients within the healthcare environment, which again is slightly tangentious politically, I suppose. So we looked at 3 questions. Well, in fact, we looked at 4, but Jackie in my group came and spoke about some of this work, and she's answered the 4th question, which I'm not going to talk about, which is about how do you define species. But the 3 questions I'm going to look at here: can we use whole genome sequencing to detect differences between isolates within an outbreak in a hospital? And if we can detect any differences, can they actually help us determine the chains of transmission? And then the 3rd question is, well, if resistance emerges, can genome sequencing tell us what's going on? So there was an outbreak in the Birmingham hospital. It was actually Selly Oak Hospital back in 2008, and all the isolates in that outbreak were indistinguishable by current typing method. So they get sent off to Colindale in London to the reference laboratory, and the reference laboratory will do some typing, and will say, yeah, they're all indistinguishable, you have an outbreak, but that's as far as they can go. And this is being written up in a paper in the Journal of Hospital Infection. We just starting to get into this, and we used 454 sequencing there. We did whole genome sequencing of 6 isolates in the outbreak, and then we detect single nucleotide polymorphisms or variants by mapping those reads against a draft reference assembly. Now, because we were doing this for the first time, we were very careful to look for false positives and remove them. We wanted to have highly trusted SNPs, and so we went through and we filtered out in the bioinformatics, but then we went back, and any SNP that we found, we went back and amplified that out of the genome and sequenced it with conventional Sanger sequencing as well. And what did we find? Well, we found that these outbreak isolates were distinguishable at only 3 loci in the genome, and these are the 3 SNPs. Now, AB0057 is a completely unrelated isolate, which there was a genome available, so we used that to polarize the change, show that that was the ancestral state at these loci. And then we here have tabulated the changes we saw. Now, when you look at that, you kind of think, well, it doesn't make much sense, does it? The M actually stands for military, so there are 4 military patients and 2 civilian patients. But if we actually map that onto kind of time and space aspects of the outbreak, we see something looking rather like this. So 4 of the patients in the outbreak were in a 6-bedded unit within the ITU, and they were in adjacent beds. 1 was out on the main ITU for some of the time, and there was 1 out on the trauma unit and had never actually been anywhere near that ITU. Now, we looked at this, and you could argue, well, it's a bit of a sledgehammer to crack a nut sequence of the genome just to get this small amount of information, but we looked at this and we tried to interpret it, and we thought, well, on the principle of parsimony, one thing we can conclude is that M2 and C2 have the same genotype. They're in adjacent beds at overlapping time, and C2 came after M2 in terms of isolation date, and therefore it seems highly plausible that a transmission event here happened between M2 and C2. Now, again, as you may well say, well, hang on, this is obvious, isn't it? It's kind of not that informative, and so what? But we were very pleased because we didn't know what we were going to get. We didn't know if we were going to get completely erratic, uninterpretable data, whether we were going to get thousands and thousands of SNPs, or whether we get every genome identical. So we were tentatively pleased with this first pilot data that we generated here. Now, those of you who are awake might say, well, hang on, you've got that same genotype down there in M4, and M4 ain't related to the others, according to what you've just told us. And we actually looked at that as well, and we went and checked the records, and it turned out that M2 and M4 were both military casualties, and they were both repatriated to the hospital through the same care pathway and arrived at the same time. And we therefore assume that they are the ones that have introduced that particular genotype into the hospital, and that they acquired it from some field station along the way, and that's our interpretation of what went on. Next, we looked at this issue of looking at resistance developing during therapy and what's going on. This was a collaboration that we went into with the HPA and with colleagues down at the Royal London. We kind of entered part-way through, so they'd already done the sequencing on 2 different platforms as a kind of, there was no particular rationale for that, they just happened to have done it that way. But we stepped in and I analysed the data for them. So they had an isolate from this patient before tigecycline therapy had been instigated, and that was susceptible. And that was 454 sequence. There was one after therapy which had become resistant and was Illumina sequenced. Now here we found something slightly different from what we found before. Here we found quite a large number, relatively large number of SNPs, 18 SNPs between the 2 isolates, and 9 of them are non-synonymous. Now, we looked at all of those and where they sat in which genes, and we were quite pleased to see that one of those SNPs sat in the gene ADES, which is part of a regulatory system which regulates an efflux pump and is known to be associated with resistance in this organism. So we were quite pleased that we'd identified a proximate cause of the resistance. But one other thing that was noticed that was rather unexpected was that in fact, in the later isolate, there were some reads missing compared to the previous isolate. Now, when we talk about the acquisition of resistance, normally we're thinking about new DNA coming into a lineage. But here we'd seen DNA lost. And again, careful scrutiny of those deletions found that one of them had actually truncated a gene called MUT-S. And MUT-S is encodes a DNA repair enzyme. And it's known that if you delete that or ablate the function of that gene, you get an increase in mutation rate. And so we believe this is the explanation of why there were so many more SNPs in this, between these 2 isolates in this lineage than we'd seen before, because this had become a hypermutator. And there are some interesting parallels here. I mean, there's always this risk that you interpret the data in a way that kind of looks good. You make a just so story, and we were we realised that that might be an interpretation, and in fact work is ongoing to determine whether it is the MUT-S that actually has changed the mutation rate, but that hasn't been completed yet. But just in the last few weeks, Sharon Peacock, working on Staph aureus and genome sequencing Staph aureus, has noted exactly the same phenomenon in a totally different bacterial species, where MUT-S, ablation of MUT-S function has led to an increase in mutation rate. And in fact, there are parallels here with what goes on during the production of cancers. So although cancers are said to be clonal, they actually undergo diversification as kind of descent with modification going on with the cancer. And a lot of that is due to early loss of DNA repair and an increase in mutation rate in the cancer. So there are kind of parallels between the way in which bacterial populations evolve and populations of cells in a cancer. Now, more recently, we've been suffering from a new outbreak of Acinetobacter. So in Birmingham, we have a situation where we get what we call serial clonal outbreaks. So we'll have an introduction of Acinetobacter into the hospital. It sputters on for a few months. In this case, it sputtered on for over a year, and then it goes away. And we do have periods of time where we don't actually see any Acinetobacter. Speaking to colleagues in London, they say, well, in their hospitals, it just never goes away. It's part of the furniture, and they can never get rid of it. It's kind of endemic. But anyway, we suffered from this new outbreak. Started in July 2011 in a military patient. This is an old slide now. The outbreak is now thought to be over, because I think we've had about a month without any isolates. But it did cause considerable problems, including some deaths during the outbreak. It was typed by the HPA, and they said, yes, you've got a unique pulsar type 27. And so we obtained 69 isolates from 29 patients from this outbreak, including multiple isolates from the same patient from different times and different body sites. Now, our colleagues in the hospital were playing games with us, and they've actually given us some isolates that aren't in the outbreak, and they knew they weren't in the outbreak, and they were just testing us out. So we went back and said, hang on, this thing's got hundreds of thousands of SNPs different from all the others. It's not part of the outbreak. What's going on there? Oh, yeah, we knew that. We're just testing you out. So far, this is actually a bit out of date. We've almost sequenced all of these isolates now. I think we're in the last run or so before we've done that. Now, obviously, the traditional shoe leather epidemiologists will say, what's all this genome sequencing for? We can work it out by just wandering around the wards and looking at time and location and working out what's gone on. Well, we kind of mapped patients who are co-located in wards and at the same time and so forth. And this is I think this is called a furball in epidemiological terms. You can't really make any sense of just from co-location data as to who gave it to whom. So we did, I say this is out of date, we've done I think about 8 or 9 MySeq runs now. We've sequenced a large number of isolates. At best, we can do this in 4 to 5 days. We didn't do all of them like that. We did batch things up as we went along, but that's the sort of timescale you're talking about. And we found 14 high-quality SNPs in the outbreak strains. We have been trying, and this is work in progress at the moment, to try and map those SNPs onto what we know about, again, time and space aspects of the outbreak. And this is as far as we've got. So one encouraging thing is that there has been an increase in the number of SNPs as you go through the outbreak. So the later isolates have more SNPs than the earlier isolates, and they have more SNPs compared to the very first isolates. There is an alternative explanation, which some people have said, well, all these variations you see are just because you're subculturing the lab, and it's just random variation in the lab. Well, the fact that there has been this kind of clock-like progression here in terms of numbers of SNPs does speak against that as an interpretation of what's gone on. The things in circles are where they have the same SNP genotype. And so we can say that 1 and 10 are related, and indeed they are. They're both military patients, similar kind of situation. They came in around the same time together. Then there's this big group here, and then sputters on a bit, and then another large group here, and then some variants here. And we can make sense of most of this. There's a couple of black arrows there where it's not quite so straightforward. Patients weren't in adjacent beds at the same time. But in both of the main cases there, we've actually been able to look at this and say, well, actually they were sharing equipment, or they actually went to the same theatre on the same day, on the same list, and that provides a plausible explanation. One of the problems with doing this kind of stuff is actually the genome sequencing is relatively easy. Getting hold of all the clinical metadata and burrowing your way into the clinical records in the hospital and finding out, okay, were they on the same list? Did they go to the same theatre at the same time? That is actually turning out to be more of a challenge. Another interesting thing, though, that we've seen here, and it's clear, this is also new, that we're actually changing our thinking as we go along and refining the way in which we interpret things, is that you can see we have a patient here, 18. It's one of the patients who's been around longest with Acinetobacter. 18A, the first isolate from that patient, is in that main complex there, but we've had a couple of other variants coming out the same patient. And so this makes it clear that in the same patient, there may be variation within the pathogen population. And that does counsel that one shouldn't be too gung-ho about interpreting these transmission events. You have to accept that it may be that there are sampling errors. So you sample two patients who each have the same mixture, 3/4 one and 1/4 the other, and you just by chance pick the majority in one and the minority in the other, and end up over-interpreting. But we're all people doing this are on the learning curve and trying to work out how to interpret data in that way. And some of these problems are going to go away when we can just sequence the whole population. So that was Acinetobacter. I'm now going to just spend a few minutes, how are we doing for time, yeah? Doing okay, talking about E. coli, and a particular nasty form of E. coli, this German E. coli 0104H4, which caused what some people call the sprout break, an outbreak associated with sprouting seeds, with fenugreek seeds. Now this struck Germany in May to July 2011, and in fact I think there's over 5,000 cases and over 50 deaths now. It was concentrated, as you can see, mainly in northern Germany. There was a high risk of what we call hemolytic uremic syndrome with this outbreak, which is a very nasty consequence of the E. coli infection. So it was not just a simple diarrhea, it actually causes end organ damage, kidneys and the brain and so forth, causes anemia through hemolysis and so on, and is life-threatening. For some reason, females are particularly at risk. I haven't yet seen a satisfactory explanation for that. Perhaps a slightly facetious explanation is that real men don't eat sprouts, but we don't know. This just shows the epidemic curve, so you can see things were at their worst in the third week in May, and then things slowly cleared up. But it did continue actually into June, and I think there were even some cases after that. And you can again see that both the gastroenteritis and the hemolytic uremic syndrome were concentrated more in females than in males, but particularly the hemolytic uremic syndrome. And that's not really been explained yet, I don't think. Now, right at the centre of this outbreak was this guy here, Dr. Holger Roeder, who works at the University Clinic in Hamburg-Eppendorf, one of the suburbs of Hamburg. And it was a truly chaotic time. They were having hundreds of patients turning up ill. They didn't have enough bedpans. They didn't have enough beds for them. They were having to open temporary wards and all sorts of things. But as you can see, he's looking at some sequences here. He is actually a molecular biologist as well, and he was thinking of what can we do to see what this is all about? What is this strain all about? How can we characterise it? And he thought, well, if we can get some DNA from it, maybe we can get it sequenced. But he didn't have that capacity himself to do that. And so what he did was what you do in that situation, you call international rescue. And those of us of a certain age knows what happens when you call international rescue. Thunderbirds are go. Well, in fact, what happened was that he managed to actually send some DNA, not on Thunderbird 2, but by conventional air freight, to the Chinese Genomics Institute in Shenzhen, BGI Shenzhen. And they had, as it happened, had just taken receipt of a new benchtop instrument, the Iron Torrent, which meant that they could run the genome through that in a matter of hours and get genome sequence. And that's what they did. And then they did something quite remarkable, which was that they released that data into the public domain without any strings attached. And this guy, Nick Lohman, who works in my group, happened to be at a bioinformatics conference, not just a bioinformatics, a conference on bioinformatics and public health in Hinxton, and noticed this. And there he was with all these other geeky bioinformaticians, and he did a blog post here saying, look, this stuff's been released. And it so happens that he was geared up to analyse that and did an assembly of that genome data, because he had learned, well, I'd just won an iron torrent, actually in a European-wide competition, and he was just gearing up to start analysing data from it. And so he was one of the few people, probably the only person in the world, that ever looked at E. coli data from an iron torrent and tried to assemble it. So he assembled it and then said to everyone else, oh, come on, let's have a go at analysing it as a crowdsourced kind of analysis. So he released his contigs back into the public domain as well. So within 24 hours of the BGI releasing the data, we had a genome assembly. Within 2 days, others rose to the occasion. We had assigned to an existing lineage. There was a guy who blogs under the name of Mike the Mad Biologist. His real name is Mike Feldgarten, but he was working in North America, and he actually did virtual MLST, kind of way of actually working out what lineage it belonged to from the genome sequence. And he actually pointed out that actually, despite all these claims about this being like the Andromeda strain, something that had never been seen on planet Earth before, it resembled a strain that had been seen in Germany a few years before. Within 5 days, strain-specific diagnostic tests had been released. And within a week, there had been over 24 reports filed on this strain, on this open source wiki, as it's called. So here's just a screen dump there. All these people communicating by Twitter and blogging and so forth. And you can't quite read it, but the slide speaks to the tremendous diversity of effort here. You've got people with Spanish names, Muslim names, Polish names, Chinese names. You've got people working in North America, Asia, even in Africa, in South Africa, people working on this. And it was really truly an international effort. And the key point was that most of these people, almost all of them, were not employed to do this. They weren't working as a public health microbiologist. They were just doing it because they were interested and they had the skills and they wanted to make a difference. Now, I was just away on holiday for a brief break, and I came back, and Nick was telling me about this by email, but I saw him face to face, and I said, well, you've been having some fun, but, you know, our world is driven by the ref. And pissing about on Twitter and blogging and stuff like that doesn't equate to a peer-reviewed research publication. So just man up and stop playing around, please. And he said, why do you have to be such a grumpy old man? And so we sat down together and thought, how can we square the circle and actually get a peer-reviewed publication out of this? And so we managed to do that. We wrote the stuff up for New England Journal with the German and Chinese collaborators. I said, well, actually, we can't just make this a genome sequence paper. We have to talk about this wonderful journey of going through the crowdsourcing as well. It did create some problems because there were quite a few people involved in the crowdsourcing, and it was already an international collaboration between people in UK, Germany, and China, and I couldn't sort of say, I want 20 new authors from the crowdsourcing. The other thing was that we had to know that the work had been done consistently and accurately by all these people. So Nick actually did heroic effort and repeated everything that everyone had done that we thought we wanted to write up in-house. And we wrote it up and we got it into the New England Journal. And it was published on July 27, 2011. As I say, I did feel a bit worried about how we were going to get the crowdsourcing people in, and we ended up creating an author called the E. coli Genome Analysis Crowdsourcing Consortium, and we named those names elsewhere at the end of the paper. I still felt a bit mean about that, and I was pleased to see when it came out in PubMed that PubMed actually recognised those individuals as collaborators, so they did get at least some recognition for what they'd done. What are the takeaway messages from all that? Well, the key point is infection's still around. You know, this idea that we should all be studying cancer and diabetes and that infection's a spent force. Well, you go tell that to people who are living in the most advanced society in the world, in Germany, you know, it's the highest standard of living. It decimated that society for a while. And there are plenty of people there now who are having to live without kidneys for the rest of their lives. Pathogens don't bother with passports, so it wasn't a new strain. Something's been seen in Germany, and it turned out something in Korea. And when you looked at the closest genome sequence strains, it turned out to be an isolate from the Central African Republic, from an HIV-positive patient there. So it's clear that these strains are moving around over large distances. And one of the interesting conclusions that we could come to was, the surprising thing was that if we look at E. coli 157, which normally we associate with sugar toxin production, hemolytic uremic syndrome, there we always look for an animal source. It's basically cow feces has got into the human food chain somehow. But this belonged to a totally different lineage, so-called enteroaggregative lineage, and therefore probably had come from a human source rather than an animal source. It's clear also that bacteria are evolving very quickly, that virulence factors are jumping around from one lineage to another on mobile genetic elements. And people in my field, and I suppose in all fields, they want to classify things and pigeonhole things, and people have got used to the idea of enterohemorrhagic E. coli, sugar toxin production, blah, blah, blah. This actually kind of broke that, because it said you could have a sugar toxin producing enteroaggregative E. coli, which kind of broke those two, overlapped those two existing pathotypes. And here are a number of the things that were new in that lineage when compared to the closest genome sequence lineage, mainly prophages. One worrying thing was that this thing was actually an extended spectrum beta-lactamase producer, so antibiotic resistance, when in fact nobody would be using those kind of antibiotics in this context. So it was kind of tooled up to be a nasty pathogen, but where that selection had gone on, we don't know. So had we created a new kind of Weltanschauung, or new Zeitgeist, to use some German terms? Well, we came up with the term open source genomics to describe the fact that you can have high-throughput genome sequencing, crowdsourced analyses, and this iterative liberal approach to data release, where everything feeds off what had gone before in a completely open way. I think one of the main lessons is that social media like blogging and Twitter, like my first response, this is all a waste of time, actually this has shown that it can augment the normal academic channels of academic discourse, and this is still a very lively subject as to whether you should be discussing things on blogs or whether you should only respond to what's in the published literature and so forth. You know, have we hooked up the wisdom of the crowd versus this kind of the mutability of the microbial masses? Have we broken the mould? Will people actually just open their lab books and let the world see? I suspect not. But I think for public health emergencies, this is obviously now the way forward. I should say public health, not public health. There is this issue about site or site. Should you put data up on your blog? Should you put it up on a preprint server? The New England Journal actually had a rule introduced by a previous editor called Inglefinger, which was that nothing could appear in the New England Journal if it had been published elsewhere. Well, all of the stuff that we presented actually had been published on blogs earlier in the year, but nonetheless they allowed us to publish it in the New England Journal. So have we shown the finger to Inglefinger? I'm not sure. We'll see. Anyway, here's us celebrating with our German collaborators. Here's Dave Rasko, who actually had a paper out back-to-back with ours. He did the same sequencing, but with the PacBio system. So on the back of all that, how are we doing for time? Yeah, I've got a few more minutes. We actually had another plan. We thought, well, let's get together with John Wayne and others in the HPA and see what we can make of the British isolates. There were 5 isolates from this outbreak made in Britain from people returning from Germany. And so he said, why can't we do something with those? And I said, well, why don't we compare the different benchtop platforms that are coming out and you benchmark them on those? And so that's what we did. And we got a nice paper in Nature Biotechnology, which has been highly cited. I'm not going to go through the technicalities of that paper. Nick Lohman's in a better position to do that. But you can see there are clear differences between the platforms in terms of cost. So the 454 Junior is basically ruled out on grounds of cost because it's orders of magnitude more expensive than the others in terms of amount per megabase. Iron Torrent has been on a steep improvement curve, so it was initially quite expensive but got very cheap very quickly, and MySeq was cheap all along. Nick did a load of stuff about error rates and so forth in the paper. I'm not going to go over that. What I said was I want a real-world readout. As a clinical microbiologist, I want you to tell me about how well it does in identifying genes of interest to me. So MLST loci or virulence genes or antibiotic resistance genes. And so we made a list of genes, a few dozen genes that we thought were of interest, and saw how the sequencing platforms combined with different assembly platforms performed. And the takeaway message here is that we don't have perfection yet, because none of the platforms work perfectly. None of them gave us a single contig, perfectly accurate chromosome sequence. But there were considerable differences. So you can see the 454 Junior with the Myra assembler only identified less than half of these genes as fully assembled genes, whereas CLC working on MySeq contigs gave us nearly 95%. Okay, the last few minutes, I'm now starting to move into provocative mode. Okay. Now, microbiology, particularly bacteriology, version 1.0 was established in the 1880s. With these two papers in particular, these are the English translations, which the ASM did a few years ago, of the original German papers from Robert Koch and Christian Graham. So Robert Koch established that it's a good thing to grow bacteria on a solid medium and obtain colonies, propagate them in pure culture. And Christian Graham said it's a good idea to stain bacteria, look down a microscope at them and look at the gram stain morphology. And clinical microbiology labs still use these technologies to this day. And if I was being provocative, I'd say nothing much has changed since the 1880s, really, in terms of paradigms. Okay, we've tinkered at the edges with a little bit of automation and a little bit of, we do the odd PCR, but you know, nothing's changed. So what I'm provocatively proposing is microbiology 2.0 is the clinical metagenomics, where instead we can take our clinical sample, stool sample in this case, we turn it into sequence data, and we analyse the sequence data, and we get to where we were with conventional approaches and beyond them through sequencing. And this is where I get really provocative, that high-throughput sequencing becomes a method of choice for diagnosing, and medical microbiology just becomes a branch of clinical chemistry or genomic medicine. So we've got a review coming out in Nature Biotechnology in the next few days. It's certainly coming out this month. I'm not sure which day it's actually coming out on. And this is a figure we put in there. So if we look at the current workflow, what's so wrong with the current techniques? Well, you have a sample collected, and then you do microscopy, which may or may not be helpful. And then you have 24 to 48 hours for things to grow, but can be even longer. And what I haven't shown on here is that there is a tremendously complex workflow because different organisms require completely different media and different growth conditions. Then you eventually do culture them, and then you have to do antibiotic testings, then you have to sequence type them. It all takes a long time. And if we could just take that sample and sequence it, then maybe we'd get to a better place. Now, I've been talking about bacteriology, and I sometimes merge bacteriology and microbiology. It's a bit like you confuse Britain and the United Kingdom and that sort of stuff. Obviously, the virologists are usually smarter than bacteriologists, and they're always ahead of us. And they've been ahead of us in this game as well. They've actually been using diagnostic metagenomics of just extracting nucleic acid from a sample and sequencing it en masse and seeing what you find in there. They've been using that to identify new viruses and find pathogens for a few years now. Not many years, but they have been doing it. But in the field of bacteriology, this really hasn't been done in this way. That people have been using amplification approaches where they amplify barcodes, but nobody's actually looked at just what you get if you sequence everything. Now, in my group, we kind of got an interest now in this, in microbial communities and culture-free diagnosis, and we have a couple of ongoing projects which I'm not going to talk about. But this, if you like, primed us to think this way. We've got an ongoing study on the chicken gut microbiome, looking at what it's in the chicken cecum and how the microbial components of that community actually involved in handling food and rescuing nutrients and that kind of stuff. And we also have this trauma centre which is a collaboration between the military and the local hospital and our university, looking at how can we diagnose infection in trauma patients more effectively and track the spread of organisms. But the main thing that we've been interested in is actually working with our German collaborators in the next round of collaboration, looking at what we've called diagnostic metagenomics. And we've written this up for the New England Journal. In fact, we decided not to go to the New England Journal with it. They said they would send it out for review. We're going to go to the Journal of the American Medical Association, to a special issue. The first draft, written here, we'd done 10 samples on the MySeq. So we'd just taken the stool sample, extracted DNA and sequenced it on the MySeq. We did that for 10 samples, and we found that we could get genome-level coverage. So here's one sample where if we align the reads from the metagenome against the genome of the E. coli 0104, we get about 20-fold coverage, 25-fold coverage evenly across the genome. There is a little bit of a smile there, but that's because when you have a bacterial genome, some parts of the genome are overrepresented because they're closer to the origin of replication than others. But one interesting thing that we found was that there is a little upswing there, and that's where the sugar toxin encoding phage is in the genome. And that's what you'd expect, because as part of the pathogenesis of infection, the release of the toxin depends on the phage actually lysing the host cell and propagating its own genome. So you'd expect to see the sugar toxin encoding phage at higher representation than the rest of the genome. We managed to recover the MLST profiles from this, and I assigned it to this ST678. And we also managed to retrieve the flagellin subtype and identified this as H4, all from no culture, just from extracting DNA from the feces. This was another one of the stool samples, and this gave us a slightly different pattern. So you can see up there is still that sugar toxin phage. There's a little uptick there in terms of coverage. But here you've got a funny looking picture. What this is actually, there were actually 2 E. coli's in that sample. And so when we align against our outbreak strain, and it's part of the core genome of E. coli, we see it overrepresented near 200-fold. Where it's the parts that are only found in the outbreak strain, we're getting 100 or 75-fold coverage. And we've been able to tease out those 2 different strains, sequencing those 2 different strains, and we've identified that one is the H4 as we expected, the other one is an H47 serotype. So we can get epidemiological information, we're nearly finished, from this, just from the genome, no culturing, straight out of the box. Now we're living in exciting times. Because last Friday, so we went to JAMA, I went to the editor who was handling this thing, and he said, you've only got 10 samples, it's a bit under spec, isn't it? I said, so we went back to our German colleagues and said, all right, we want to do about 50 samples now to spike that particular criticism. We did argue with the guy, hang on, have you not seen this thing in Science Translational Medicine where they sequenced 2 newborn genomes, N equals 2, and it made all the headlines, and we're doing something similar, but okay, just to shut you up, we'll do another 5 times as many samples. And we've done some of the samples at 10-fold deeper coverage. So this has gone on to the High-seq instrument at Illumina. It's a new High-seq 2500. It's gone on 2 times 151 base pair in so-called rapid read mode, which is about 24-hour run time. So they were put on the machine on Monday, and overnight the data has been streaming off into this place called base space. So Illumina have just set up this cloud where they put all the sequence data from, you can put sequence data from runs. And so Nick in my group, as you can see, he's a real enthusiast. He's actually blogged on this. He's got so excited, he kind of almost went into. In fact, he did actually say on Twitter that I think it was not last night, the night before, when this first started streaming through, that he was watching this rather than the television because it was so exciting watching the numbers change. Anyway, some people do get so enthusiastic and so geeky that that's what they do. But we've got 48 samples. We're getting, in fact, that's not quite right. It's 180 gig of data. And we reckon that, because we've gone 20 gig on some of these, that this is the deepest that any human single stool sample has ever been sequenced. So it's kind of exciting. Nick has got the task of having to write all this up to analyse it all in probably next 3 or 4 days, and then I've got to write it up. And we've set the goal of submitting at the end of next week, because this special issue comes out early next year. We need to get in. We're supposed to get in today, but we've had a week's grace. So, I hope I've convinced you that genome sequencing really does bring all these advantages, and that benchtop sequencing is in a good place. Platforms have defined strengths and weaknesses. I hope I've excited you, but I have to end by saying you ain't seen nothing yet, because there are things out there which are even more wonderful than what we've seen up till now. So Oxford Nanopore have announced earlier in the year a platform which includes a USB stick like this. You can pipette your sample into it. You don't have to do any library preps. Sequence is read, streams onto your laptop in real time. And this thing costs $500. That's the hope. The hype, why do I say hype? Well, because nobody's seen any data from this platform. No independent group has ever used any of these instruments. And there is just a small question of whether they've hyped this up to get money out of venture capitalists, and it's all just vaporware. I don't know if we've got time, but I've got a little video here, if you're interested, to show you that how cool that technology is.
This model shows a typical nanopore made from protein. You can see that at the core of the protein is a hollow tube that is only a few nanometers in diameter. Oxford Nanopore designs and manufactures bespoke nanopore structures for a range of applications. In nature, nanopores form holes in membranes. In Oxford Nanopore's system, the nanopore is inserted into a membrane created by a synthetic polymer. This membrane has very high electronic resistance. Here you can see a nanopore piercing a single hole in a membrane made from synthetic polymer. A potential is applied across the membrane, resulting in a current flowing only through the aperture of the nanopore. Single molecules that enter the nanopore cause characteristic disruptions in the current. By measuring that disruption, the molecule can be identified.
Sorry about the cheesy music, you always get that in these promotional videos.
In strand sequencing, an intact DNA polymer is sequenced as it passes through the nanopore. Here you see a DNA enzyme complex approaching the nanopore shown in blue. The enzyme shown in green is designed to ratchet the DNA strand through the nanopore one base at a time. The enzyme binds to the end of a double strand of DNA and unzips the double strand to form a long single strand which it feeds through the nanopore. As the DNA strand moves through the nanopore one base at a time, a characteristic disruption in current is created by the presence of particular combinations of bases in a particular part of the nanopore. Because these disruptions in current are so specific to the different combinations, this information can be used to determine the order of bases on that DNA strand. There is no deterioration of accuracy as the long DNA strand is sequenced. By preparing the DNA so it has a hairpin structure at its end, the system can read both strands, that is, the sense and antisense strands of the DNA. This gives advantages in data analysis. In exonuclease sequencing, a nanopore is coupled with a process.
That just gives you a feel for things. As a salesman, I have to tell you that there are, stocks may go up and down. There are some challenges and caveats here. We haven't solved everything. I've listed some of them there. I think one of the biggest challenges is that clinical microbiology remains a very conservative discipline, and although we might be able to do this in the university environment, actually moving it forward into the real world, and communicating with clinicians, telling them that they've isolated Stenotrophomonas multifilia means that going, oh, hey, what are you on about? Trying to tell them about the genome sequence of the organisms they've isolated. Well, that's going to be a challenge as well. The other key point, of course, is that technologies don't always replace each other. Radio survived alongside TV. You might argue, well, TV is clearly a superior technology. I suspect, culture will, and traditional approaches will still have a place, and it's just going to be interesting to come back in 10 or 20 years and see where we are. Perhaps one way to look at it is that the promised land is in sight. Here's Moses looking across the promised land, that vision of all those sequences, but there's still many, many rivers to cross before we get there. That's a point I made in a piece we did in genome medicine. And that's me finished. I just have to thank all the people who've done the work, particularly Nick Lohman. Here's one of his home cloning experiments and his wife. And here's Kristana Konstantinidou, who's actually been the person actually hands-on the instrument doing the sequencing. So the two of them together have provided a perfect team, really, for this. And lots of other people as well. I haven't got time to name. If you're interested, you can look at our blog or our Twitter feeds or YouTube. That's it. Thank you.
Eyes open, they weren't open beforehand to what possibilities exist in this technology.